Hello, today I'm going to tell you about a novel treatment for acute kidney injury. How big of a problem is this in the United States? Well, every year we have 36 million admissions to hospital, and approximately 7% of these people will develop acute kidney injury, leading to 2.5 million acute kidney injury episodes per year. This clinical scenario is caused by decreased renal perfusion, which can occur during surgery, with sepsis syndrome, hypotension, or radiologic procedures. And we believe that the technology we're going to show to you today can be applied to approximately 15% of all the people with acute kidney injury, leading to over 400,000 patient treatments per year. This leads to an addressable market of about $1.6 billion per annum at a price point of $4,000 per treatment. So how do we recognize acute kidney injury? The diagnostic criteria in the clinic is to monitor patients' urine output. We notice that the urine output begins to decrease, and there's a concomitant increase in the patient's serum creatinine. The current plan of therapy is to observe the patient daily and to monitor for life-threatening complications which necessitate the need for dialysis therapy. It should be noted that dialysis therapy does not intrinsically reverse the acute kidney injury. It merely prevents you from some of the most onerous complications of acute kidney injury. So the outcomes are rather dismal. There's a fourfold increase in length of hospital stay, a fourfold increase in likelihood of transfer to the intensive care unit, and a sevenfold increase in the likelihood of death. Notably, the incidence of acute kidney injury requiring dialysis has been increasing over the past 10 years, and it's almost twofold that of what it was 10 years ago. So what is our proposed solution to this? Well, it's really quite simple. In an interventional radiology suite, a radiologist would pass the catheter through the femoral vein up into the renal vein, and there dilate a balloon and do a rapid hydrodynamic pulse of fluid, which is basically a physiologic saline solution. And the idea behind this is that we would deliver the fluid in such a way that would reproduce the kind of delivery of fluid that we've seen in laboratory animal studies. There would be a controller system that would memorialize this delivery of therapy, and that is the source of both the Purdue and IU technology. If we examine what occurs when we do this sort of thing in laboratory animals, you induce acute kidney injury at day 0, and we're measuring serum creatinine versus the time after injury as our marker for renal function. If we intervene 24 hours after the injury, which is a clinically useful time period, at the time we do this delivery, we see a stepwise decrease in the serum creatinine. In contrast, if we deliver fluid peripherally, you see that the serum creatinine continues to trend up for an additional day and then starts to march downward. Furthermore, we have found that tissue histology scores show an improvement in tissue injury, and this is all due to clearing of capillary occlusions by aggregated red cells. We also found that there's a dramatic decrease in inflammatory immune cell infiltration into the kidney, which suggests that the long-term injury process that is initiated by acute kidney injury will be dramatically reduced. So the basic technology is a patent pending catheter, which is a balloon catheter with a pressure transducer and pressure sensing system in the catheter, and a large bore so that you can deliver fluid in a rapid infusion, and a pump controller in which a physician in an interventional radiology suite would be able to easily memorialize what we do by simply pressing the start button. This is a fully programmable unit, and it also has safety features which include an emergency interruption to the procedure should something go amiss. We're currently in the seed round phase of this. We think we need somewhere between $400,000 to $500,000 to complete a non-GLP feasibility study in pigs. We already know that the procedure is feasible in pigs, but we need to confirm safety and efficacy. We have Gen 1 catheter development well in progress. We need some regulatory and clinical strategic planning and an expand the IP portfolio. If you're interested in this, we're interested for a investment of 400 to 500,000. Interested investors should contact Tyson Rugerstein at IU Tech Transfer. We're interested in either an industry collaboration or to start a biotech startup. Thank you for your attention.
All right, so the technology I'd like to talk with you all about today is how we can use antimicrobial peptides as a scaffold for the design of new therapeutics. And the peptides we decide to target are the bacteriocins. They're specifically the antimicrobial peptides of bacteria. And bacteria will use these to compete with other bacteria within their environment, whether that be allowing them to establish a new community or fending off invading pathogens that might be around them in their natural environment. And we want to use this to address the problem of antimicrobial resistance. So it's been projected that in North America alone, there will be up to 300,000 deaths per year from antimicrobial resistance alone. And this has created a future antimicrobial market that's valued around 45 billion US dollars. And not only is this creating a market for the sale of new antimicrobials, but it's also causing direct societal and healthcare costs. Healthcare costs in the range of 20 billion US dollars and more societal costs in the range of 35 billion US dollars from the loss of time at work. These types of peptides have traditionally been used as food preservatives. So just kind of a case study here, nisin was a bacteriocin that was first discovered in 1928, and it wasn't actually approved for use by the FDA until 1988. So there was a large 60-year gap there between the discovery and the utilization of this product in the market. And it's mostly been used as food preservatives. There's a few companies in Denmark specifically that will use these types of bacteriocins to preserve food, mostly dairy, meat, and cheese products specifically. But this limited use is really fallen because of the limiting factors in the utilization of these types of compounds. They have a very limited spectrum of activity, meaning they'll only kill certain bacteria. They won't kill a lot of different kinds like general antibiotics. They require very specific production conditions. Bacteria don't just tend to make these because they want to. They'll usually only make them when they feel like they have to. And really finding that condition can take a lot of time and utilization, pouring in a large amount of R&D hours. These also tend to be rather large peptides, which can make them hard to synthesize chemically. And in addition, they often require a large biosynthetic machinery and other modification enzymes that are needed to turn this precursor product into the final active product. So to kind of get around this, our lab started looking at how we can use bacteriocins from plant-associated bacteria to actually kill other plant pathogenic bacteria. So we developed a rapid platform for the development of bacteriocins as scaffolds for the design of novel antimicrobial peptides. So we first start off by genome mining a bacteria of interest for a bacteriocin that might be amenable to future development. So this allows us to quickly identify any novel antimicrobial peptides and removes the need for bacterial characterization, as well as identifying the production conditions necessary. From that, we identify a minimal unit of those bacteriocins that we believe to target the bacterial membrane as a mechanism of killing. This allows us to generate small peptides from the larger peptides. Because these things are constructed of the 20 naturally occurring amino acids, they are amenable to genetic engineering and expression in heterologous hosts. And finally, we can extend the spectrum of activity by using a library-based optimization approach. So this will allow us to enhance the antimicrobial abilities of these peptide scaffolds, making them better at disrupting the bacterial membrane itself. So the data I have here are some minimal inhibitory concentrations. So this is a measure we use in microbiology to determine the strength of an antimicrobial. So the lower the minimal inhibitory concentration, the better that compound is at killing bacteria. So under here, under strain, we have a lot of bacteria: Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and E. coli. These are all bacteria that can cause nosocomial infections. These are also bacteria that are members of the escape pathogens, or bacteria that are very often resistant to antimicrobials and will pose a great human health threat in the coming years should antimicrobial resistance remain unchecked. So in the first red box, SynSAF-P1, that's our starting peptide. It's not very active against the bacteria used. However, in the final row, final column of that table, SynSAF-P96 is our optimized peptide. And what we notice is we're able to get that MIC value much, much lower through our approach, meaning we've enhanced the ability of that peptide to disrupt the bacterial membrane and kill the bacteria of interest. Towards the bottom of this table, you'll also notice two bacteria, Xanthomonas oxanopodes and Pseudomonas syringae. These are two plant pathogens. And we notice that our peptides are also very good at killing these types of pathogens as well, which could highlight not only their use in human health settings, but also their use as in agriculture to protect crops. I also want to look down at this table down here. We've applied this approach to different peptide scaffolds from different bacteria as well. And in the red box here, I just have Staphylococcus aureus USA 300 highlighted. Staph aureus is a MRSA strain, a methicillin-resistant Staphylococcus aureus strain. It can cause a host of skin and wound infections, both in the community and in hospitals. And often they are very, very difficult to treat with antibiotics, meaning there's a great need for alternative therapies. And I really want to highlight SynLarv P1 and SynLarv P71 in this table. So SynLarv P1 is our starting parent peptide, and we notice that it's not active at all against the Staphylococcus aureus strain with a very, very high MIC above 384 micrograms per mil. However, upon utilizing our optimization approach, we're actually able to get that effective concentration down into the nanomolar range of about 0.75 micrograms per mil, meaning we've greatly enhanced the ability of this peptide to kill our target bacteria, which is very exciting. And we could compare this to the MICs available against this strain for vancomycin and linezolid, which are two point-of-care antibiotics for these types of infections. And what we see is that our peptide scaffold and our approach has actually designed a antimicrobial peptide that is more effective than vancomycin and linezolid in these lab-based assays. So this work is still ongoing, and we have a lot of extra data that we could show. If you'd like to see any of that data or reach out to us about licensing or collaboration, please feel free to address Dr. Sean Lee, who is the PI and one of the inventors on this project, or Richard Cox at the Idea Center at Notre Dame, who's the Director of Business Development. So yeah, thank you very much, and I'm grateful for your time.
Radivir was started a little under a year ago out of Purdue University in Dr. Phil's lab, based on some technology that a graduate student had written his thesis on, focused on influenza. And when the pandemic hit, the thought was to apply that same technology, but to coronavirus. The work that was done on flu was received very well. It was eventually published in Nature magazine. And so the thought was to transfer that success, if you will, to see if we could address coronavirus. And to date, we've been very successful in doing that. There probably isn't much of a need to go into detail of why it's important to develop a therapy for coronavirus. The data here is very familiar to most, if not all of us, in terms of the impact, both in terms of human suffering and financial and just life in general. Total cases have topped over $100 million worldwide, and deaths over 2 million. And what's interesting here is it's not slowing down. Active cases is a quarter of all those that have happened to date. There has been progress made, fortunately. We all know about the virus vaccine, and a lot of work has been done on therapies. First, repurposed drugs for the use in coronavirus, and a success story was had with remdesivir, which was made by Gilead. And the impact that it's had for these companies that have introduced these technologies has been great. When remdesivir had success initially, the market cap of Gilead increased almost $20 billion based on that. When Moderna and also Pfizer came out with their vaccine, Moderna in particular, the market cap, which originally was a $20 billion market cap, increased $40 billion, 3 times what it was before because of the potential of their vaccines. So there's plenty of reward for those that develop a therapy and a vaccine for the coronavirus. So how does this technology work? In Phil's lab for the last 40 years, he's designed molecules that target pathological cells, represented here by this blue circle. We have developed, Radivir and Purdue have developed a targeting molecule that binds to the spike protein found on coronaviruses and also the host cells. It binds at a very unique spot. It is at a spot that has not really mutated to date. It is a spot where it can be found on spike proteins for MERS and SARS and potential future viruses. The red figure there is then representing the payload that's attached to this homing molecule. This payload is a beacon or a signaler to the body's immune system, which is represented in the lower right. It calls the body's immune system to come and attack the virus and the virus host cell. So what's the advantages of our technology over vaccines and over other therapies in the process of being developed? Most of them are antibodies. First of all, the basic technology is a small molecule. It is not large, which means that it's safer. It clears the body quicker. Many of you have heard of the concept of cytotoxic storms. These are caused by the immune system overreacting and causing organs to shut down and have trouble. With a small molecule, our technology gets to the affected cell and clears and can be managed. It's safer. It's faster to work. It gets there. And so you can take it and feel better soon. It is not preventative like a vaccine, but is much more cost effective because it's easier to make and it's mutant resistant. And as I mentioned, it can treat future viruses. So what are we asking for? We've raised $1 million from government grants like the NSF and from founders like myself have invested. Purdue University and a local VC have invested to reach $1 million. We need $500,000 more in order to develop the drug to the point where we can test it in animals required by the FDA and eventually get into humans, which is scheduled for next year. If you're interested, we'd love to talk to you and would be happy to tell you more of the story. Thanks for your time.
Light-activated treatment for triple-negative breast and prostate cancer, presented by Professor Maden F. Isaac Lam. The technology that I will be presenting today is in the field of photomedicine, in which light is used as anti-cancer treatment. Triple-negative breast cancer is a type of breast cancer without hormone epidermal growth factor, estrogen, and progesterone receptors. 15 to 20% of breast cancers are TNBC. They typically are more aggressive, more difficult to treat, and more likely to spread and reoccur. Thus, there is an urgent need for novel therapy due to non-specific treatment regimen. 2018 US statistics showed that breast cancer is the leading cause of death for women 20 to 60 years old, and prostate cancer is the second leading cause of death for men of all ages. This technology is also referred to as photodynamic therapy or PDT. It involves light and natural products such as chlorophyll and vitamins for targeted delivery into cancer. The novel drug therapeutics I developed are composed of chlorophyll derivative bonded to vitamin B7 or vitamin B5. These compounds absorb visible light, triggering formation of reactive oxygen species, killing cancer cells. The drug therapeutic is introduced intravenously in the body within 24 to 48 hours, localizing in the cancer cells. Tumors are then exposed to light, shown on the right side, delivered through thin, flexible fiber optic cables. This laser light penetrates tumor tissues and activates the drug. Because the drug is inside the cancer cells, only the cancer cells are destroyed, avoiding healthy cells. This technology aims to target vitamin receptors overexpressed in cancer cells, as shown in the cartoon in the right side, to localize light-activated drugs in the tumor cells. PDT can also be used as a combination therapy to eliminate remnants of the cancer. The compounds are developed, have low toxicity, are effective at nanomolar concentrations against triple-negative breast and prostate cancer cell lines. So I took care of the women, the men, and everybody. As shown on the right side, this is the image for early lung tumor before PDT, and 24 months post PDT. This is the image. It's clean. These compounds are also easy to manufacture. Last year, Biospace Market reported that the compounded annual growth rate for PDT in photomedicine for 8 years is about 17%. In the US, the estimated fatality for triple-negative breast cancer this year, 2021, is close to 9,000. For prostate cancer, it's about 34,000. And for cancer of the respiratory system, such as the lungs, it's about 137,000. That's quite a lot of people. So, we really have a very serious problem to solve so that we can save all these people who will be sick with cancer of the breast, prostate, and the lungs, especially in this time of pandemic. The table below shows a comparison of the current cancer treatments: PDT, chemotherapy, radiation, and immunotherapy. Photodynamic therapy has very good specificity, low toxicity, and low cost. So photodynamic therapy has a very good potential in its application to cure cancer. If anybody in the audience is interested in learning more about this technology, please contact me. My email is shown below. And thank you everybody for listening. Bye. Oh, wait. Stay healthy, be safe, wash your hands, wear your mask, and maintain social distancing because these little bugs are still around us. Hope to see you soon in good shape. Thank you.
Hello everyone, my name is Sandro Matosevic, and today I will be presenting our new technology on genetically engineered natural killer cells as immunotherapy of glioblastoma. This is a technology we have been working on for a few years and are really excited to be sharing with everyone, as we believe not only that there's a tremendous need for it, but in the tangible and real potential to make a real difference for patients that need it and currently have no other options. Glioblastoma is the most deadly brain cancer with an average survival of only about 15 months. This survival is despite very aggressive treatments. Patients with brain cancer go through immediate surgery, chemotherapy, and radiotherapy, often in combination and often over multiple rounds. It is such an aggressive cancer that almost every diagnosed patient dies, primarily because there have been no improvements in survival over the last 2 decades. These dire statistics illustrate really how desperate the need is for new treatments. In the last few years, immunotherapy based on genetically engineered immune cells has emerged as a really transformative, exciting new modality for treatment. But so far, none of the developments in CAR T or similar therapies have resulted in any change in patient survival. At Purdue, we tackle this. In terms of size of therapeutic market, about 13,000 people are diagnosed with brain cancer or glioblastoma every year. The survival rates, as illustrated on the graphic on the right side of this screen, are dismal. Barely 6% of patients survive 4 years after diagnosis, and even 1 year after diagnosis, only 38% of patients manage to survive. Because no treatment exists to stop or cure this cancer, new treatments are desperately needed. In recent years, immunotherapy has emerged as a potentially transformative treatment for this cancer, but yet has resulted in no changes in survival. Our technology is based on the first of its kind genetically engineered immune cells to treat glioblastoma. These cells are called natural killer cells. They're potent, safe cells that have an innate ability to kill cancer targets. By engineering these cells, we equip them to be more aggressive at recognizing and killing glioblastoma cells. The diagram on this slide, on the right side of this slide, illustrates the multifunctional genetic platform in action. Typically, cell engineering approaches fail because they usually target one cancer marker at once. This causes the cancer to return or relapse, patients relapse, and in an altered form, or escape recognition by expressing a new marker. We have addressed this. Our technology is different because we target 3 markers of brain cancer at once. They are called GD2, NKG2DL, and CD73. So we really limit the cancer's ability to escape killing. This results in a significantly improved immunotherapy. Since publishing this technology, we have, just a couple of months ago, we have received over a dozen emails for patients and advocates for this therapy, which really highlights the interest and the desperate need that a lot of patients have for new treatments. These steps in manufacturing of this technology are illustrated on this slide. This technology manufacturing involves engineering immune cells that are retrieved from donors. The engineering takes place in a lab. These engineered cells are then dosed into recipients or patients with brain cancer for treatments. The beauty and uniqueness of the approach is that NK cells are very safe, so that they do not require the patient to also be the donor, so they can be matched among different individuals, which really makes the manufacturing a lot cheaper and a lot safer. The data shown on the right side of this slide are some of our in vivo studies from our published data using human patient brain cancers. We've received this from a hospital at IU. We tested them against our technology. We've seen some really remarkable response against these very treatment-resistant brain cancer cells. The advantage of the technology are illustrated in this slide. Our technology is safe. It is the first human-engineered NK cell therapy for glioblastoma that has been developed and published. It is both safe and economical. Patients don't have to provide their own cells. There's no graft versus host disease, unlike treatments with CAR T cell therapies. And it's multi-specific, meaning it really limits the ability of the brain cancer to return. Some of the advantages and the advantages are of our technology compared to other CAR T at both City of Hope and Harvard are illustrated here, while the other CAR T therapies are very specific and potent. They don't address the ability of recurrence, which ours does. And also ours is much more safe for administration into patients compared to CAR T cell therapies or checkpoint blockade, which have much higher side effects. As next steps, we're looking for investors and partners to both clinically develop this platform and to further advance it into the translational treatments of brain cancer. We really believe there is a huge translational potential for engineering immune cells, and we're excited to be the first ones to bring such a therapy hopefully to patients. Thank you very much.
Hey everyone, my name is Prakash. I'm the Associate Director of Research for the Institute of Precision Health at the University of Notre Dame. Today I'm going to talk about another technology of ours where we use a novel nanoparticle-based modulator to control immunotherapeutic action on cancer cells. More specifically, we use this to depersonalize CAR T cell therapy so that we can use a single CAR T cell line and target cancer cells from which have different origins. So we can target cancer cells such as ovarian, prostate, or breast cancer, all by using a single CAR T cell. As far as the market need and problem validation, this has all been confirmed. The problem has been validated. Cancer is a problem, and there is a market need for better therapeutics. CAR T is already available in the market thanks to Juno, Kite Therapeutics, and also Novartis. In a very simple form, you take a patient's T cell, you insert a gene wherein that expresses a receptor, which is an antibody to a cancer biomarker. And once this antibody on the CAR T cell binds the cancer cell, you have activation within the CAR T cell to express cell-killing cytokines. And this is basically how it finds the cancer cells in the patient and also kills the cancer cells. There are two problems, of course. One was when they tried to translate this to solid tumors, there was off-target toxicity, which resulted in the patient's lungs and heart being targeted by these CAR T cells and resulted in patient death, which was a very bad outcome in clinical trial. The other one, of course, is because of the personalized nature of this therapy, there is a significant bottleneck when it comes to scaling of manufacturing, that it takes nearly at least 21 days for these cells to be taken out of the patient body, genetically modified, expanded, and then re-injected in. And since many of the patients are already in an advanced stage as far as the cancer prognosis goes, this is 21 days that many of them cannot afford to wait. And in case the therapy doesn't work, they really do not have a chance at a second shot. So target markets, we've chosen to highlight breast cancer. Of course, any cancer with a solid tumor is what we are going for. And we've also shown that it works against prostate cancer cells and ovarian cancer cells. So how does this work? I can't go into too many details about this, but I would strongly encourage you to get in touch with the tech transfer department. Essentially, we use a nanoparticle-based switch system such that we can retarget a single CAR T cell to multiple types of cancer cells. And by doing so, we can modulate cytokine release by the CAR T cells to mitigate cytokine release syndrome. And we can also modulate CAR T localization, which in turn allows us to prevent off-target toxicity. So as far as what you're looking for, we're looking to accelerate product translation from the lab to the bench, from the bench to the bedside. In order to do so, we think industry partnerships and collaborations, which will help us validate our platform in preclinical studies, will be crucial, especially in bumping up our TRL level from the current 3 to 4 to 5 to 6. And once we do that, I believe we can get funded through private or government sources to further bump the TRL up to 9, from which we can seriously start thinking about clinical studies so that this is actually translated to the market and helps real people with real problems. As a technology comparison, ours is very agile. It's depersonalized and no more expensive than current state of the art. I've only chosen to highlight Kite, Juno, and Novartis. Ours is especially agile because we have managed to overcome the manufacturing bottleneck of 21 days. Ours will only take 2 days to make it patient specific. And again, it's not personalized therapy. We come up with a novel workaround for that. And it's for this reason, like even if we fail in the first round, I think we'll get multiple attempts to find the right dose or the optimum treatment strategy to get the best results for the patient. So again, please feel free to reach out to our tech transfer department. We are really excited about this technology. And I think there's some real world implications, real world uses, real world benefits. And we really would love to hear back from you, your feedback, your questions, opportunities to collaborate. Any questions you might have, please feel free to reach out to me or to our tech transfer department. And with that, I'm going to stop this presentation. And we look forward to hearing back from you guys. Thank you.
Hi everyone, this is Xiaoping Bao from Purdue, and welcome to my Purdue Technology Showcase. And my technology today is off-the-shelf hematopoietin and immune cells for target immunotherapy. As everyone knows, cancer remains as a global challenge, and currently hematopoietin stem cell, short for HSC transplantation, and chimeric antigen receptor, short for CAR T cell immunotherapy, are FDA-approved curable treatment for various blood cancers. To fill this market need, reliable sources of high quality, off-the-shelf HSC or CAR T cells are needed. In terms of the market size, cancer immunotherapy market itself was more than $120 billion by 2026, and the HSC transplantation market will continue to grow by 2024 with an annual growth rate of 6.2. And as a growing market highlights the importance to secure reliable cell sources for potentially off-the-shelf cellular therapies to treat this disease. And to fill in this gap, our lab is invented a technology to use human pluripotent stem cells as a study material to make off-the-shelf HLC and CAR immune cells. And the technology one we want to share is hematopoietic stem cells of production of these cells from hematopoietic stem cells. As you can see in this slide, the top one are a platform already implemented in a company called Stem Cell Technology. And the bottom one is pretty similar, but we have a higher efficiency, we have a high yield, and we also are much cheaper, $600 versus $80, as you can see here. And more importantly, our cells are more homogeneous and higher quality by in vivo zebrafish and mouse data we collect. As an example shown here is the transplantation of human HSCs into zebrafish model. As indicated by red and arrow, you can see human HSCs can home and engraft in zebrafish, and they can also rescue bloodless zebrafish as compared to the neuron control. And the second technology is we invent a platform to genetically engineer stem cells with CRISPR-Cas9 to make a stable CAR HPLC cells and use the previous hematopoietic stem cell technology to make hematopoietic stem cells, as well as later on the off-the-shelf CAR immunotherapy, including CAR T, CAR B, CAR NK cells, and many other types as well. And as compared to the traditional CAR immunotherapies, we have a much higher efficiency to express CAR, about 30 versus 100. And also, these are off-the-shelf versus months production as compared to traditional approach. And we also have higher cell yield and high anti-tumor cytotoxicity. As you can see here, we engineer the stem cell with a brain tumor specific CAR, and as compared to the non-brain tumor, also another normal stem cells H9, our cells NK CAR NK cells differentiate from stem cells, can be more efficiently killing the brain tumors. So as a brief summary, we present here two technology, and compared to the leading biotechnology companies in the field, our platform in Purdue is faster, stronger, and inexpensive, highlight their potential to fill in the market. And by next step, we are looking for companies like Stem Cell or similar to manufacture and develop differential kits. And also, we are interested to collaborate or license our patent to potential large or preclinical studies, as well as clinical trials.
Hello, my name is Tim Corson. I'm an associate professor in the Department of Ophthalmology at IU School of Medicine. I'm excited to share with you our novel small molecule therapies for eye diseases. New therapies are sorely needed for neovascular eye diseases. These are diseases characterized by abnormal blood vessel growth termed neovascularization at the back of the eye. And this is something that is associated with major causes of blindness, like wet age-related macular degeneration and proliferative diabetic retinopathy. There are some existing drugs for these diseases, but all of them target the same pathway, which is vascular endothelial growth factor, or VEGF signaling. These drugs are not effective in all patients, and they're biologics, so they have to be injected directly into the eye. creating a risk for infection and, of course, patient discomfort. Therefore, development of topical or oral agents is very appealing. Together, these neovascular eye diseases are a major public health problem worldwide. It's estimated that there are 20 million wet AMD patients worldwide with a current market of about $10 billion. There are even more diabetic retinopathy patients, over 90 million worldwide, with a current market of $8 billion. If you look at the graph on the right, these are estimates of the projected numbers of patients in the US specifically over time, and you can see that this is increasing quite dramatically. These are data from the National Eye Institute. So to address this unmet need, we have novel technology. We have developed small molecule therapies that can be used for ocular neovascularization. Our compounds target novel enzymes, soluble epoxide hydrolase, which is involved in lipid modulation, and ferrochelatase, which is involved in heme biosynthesis. Our compounds include new inhibitors, including SH11037, which blocks the function of soluble epoxide hydrolase, and SH17023, which blocks the function of ferrochelatase. We also have a repurposed agent, the old antifungal drug griseofulvin, which was known to be a ferrochelatase inhibitor and which we have shown is effective at blocking neovascularization in animal models. Because these compounds are all small molecules, they have the potential for oral or topical, that is eye drop delivery. In a recent survey study that we conducted, more than 3/4 of patients indicated that they would prefer eye drops to the current intravitreal injections right into the eye. To the right of the slide, I have one data figure showing the effectiveness of one of our compounds, SH11037, in a model of choroidal neovascularization, which is what's seen in wet AMD. The neovascular lesion is shown in red in the vehicle-treated eye here, in the eye treated with the anti-VEGF antibody that's equivalent to the standard of care, and with a single intravitreal injection of our compound here. Below in the graph, you can see the volume of the neovascular lesions. So smaller bars represent a better therapeutic response. The standard of care works well, and there is a dose-dependent decrease in neovascularization with our small molecule. We've also shown synergy between our compound and the existing agent and efficacy as an eye drop, and similar effects in this animal model and others with the other compounds that I mentioned. We feel that our compounds have several advantages over the approved anti-VEGF drugs, which are all biologics, small molecules being developed by competitors, both startups and big pharma, and our 3 compounds developed at IU shown here. Of course, all agents are effective in animal models, but the anti-VEGF biologics cannot be delivered by eye drops, and virtually none of the competitor compounds are being delivered by eye drops either, whereas we have shown successful eye drop delivery with our agents. Our compounds act by a novel mechanism. The existing anti-VEGF drugs, by definition, act by a mechanism that is not effective in all patients. And several of the competitor technologies are also pursuing the same mechanism, although some are exploring others. The biologics are very expensive, whereas small molecule approaches are likely to be cheaper. So we feel we have several advantages. Our next steps are to found a startup in 2021 to conduct pre-IND studies for these compounds as we develop them further. At this point, we are looking for a CEO for the company, investors, and potential industry partnerships to move this exciting technology forward. Thank you for your interest.